Okay, continuing on with this part of our lecture, we will be discussing on RNA-seq. So in the previous part, we discussed about microarray, how do we do microarray, and what are the questions microarray seeks to answer. Now, microarray is considered a relatively old technology right now. Because actually technology is very fast in evolving. Now, RNA-seq is one of the newer technologies, which is an improvement on microarray data. So RNA-seq or RNA sequencing, here you have, just like microarray, you have here cells that we will be comparing. So cells under two conditions. So either diseased or healthy, or here in this example, a normal skin cell and a cell with a tumor, or rather a tumor cell. So these are the samples these are your samples, your cell samples. So you will extract your RNAs here, and then you will attach, or since it's an RNA, like this is messenger RNA, they have a poly A tail as part of the post processing. So these RNAs will be fragmented. Basically, you will do sequencing. So you will sequence this RNA that you extracted. So unlike the microarray where you will just do reverse transcripting and then hybridize those cDNAs to chips, here you will be actually sequencing the actual RNA transcripts. So you will have, just like in this, here you have multiple colors, actually 4 different colors because this is a sequence. So we are, instead of looking for fluorescence probes that are overwhelmingly red or overwhelmingly green, that stands for either of the conditions, here you have sequencing, and the colors you are seeing here are not the number of concentrations, but rather what are the bases that are being added to the sequence, because we are actually sequencing these RNAs. Now, you have your, after sequencing them, we will then rearrange them. So we will map these sequences to genomes and do analysis. So this is made possible. Microarray is very simplistic compared to this one. This is a bit complicated because this is made possible with our computer systems right now, which can actually process lots of information. So this sequence will be done for downstream analysis. So how do we then compare? So basically, by sequencing the RNA, we will know exactly what are the sequences of the genes and what are their exact, or basically objective concentrations. So unlike the microarray, which we have here a comparison between A and B, and then which A is overwhelming. So basically we are comparing. So, and the baseline is a bit difficult to determine here because it's a comparison between the two. Here we can actually absolutely determine or measure the exact concentration of a certain type of mRNA, based of course on their sequences. So not only are we getting the sequences of the transcriptome, we are also getting the concentration of each RNA in the transcriptome. So it's a pretty powerful tool. So what are the advantages of RNA-seq over here comparison with microarray? So basically, as I said, it's the absolute measure of transcript abundance, because microarray, as I said, this is just a relative measurement, because we are comparing is condition A more than condition B. So basically, it's a relative condition, more or not so more. Parang basically, is it more or less compared to A? But here you are saying that this is the exact concentration of your gene. So this is say 3 nanomolars, basically something like that. So you have an absolute measure. So, and then we have a de novo transcript assembly. So sometimes the de novo transcript assembly is important because what if these RNA transcripts, we have single SNPs here, so single nucleotide polymorphisms in these RNA transcripts that is visible. So we have here overwhelmingly, for example, especially for tumor and cancer cells, they are known to mutate. So not only will their genes have different, will they have different transcriptomes, will their genes be different, expressed differently, but also sometimes the genes have been mutated. So we can actually identify them. So we can identify de novo transcript. This de novo transcript assembly allows us to actually identify alternative splicing events. So what is an alternative splicing event? So alternative splicing, if you remember from the RNA processing in molecular biology, so this is where you have a gene, the raw transcript from a gene, and then when you process it, because we splice the introns away out of this transcript to retain the exon. So sometimes we have alternative splicings. So we cut off some parts to get protein A, and then from the same gene we can cut off a different part to get protein B. So from a single gene we can get two or more different types of proteins. So basically these are alternative splicing events, and this is difficult to see in microarray because we are using the raw transcript, and we are just depending on the primers that you have been using, you will not see the different arrangement of the exons in this RNA if it's not covered in the by the primers that you use during the reverse transcription and amplification stage in microarray. So here is a general workflow of gene expression data analysis. So it's a pretty large graph, so I actually cut it off. So you have experimental data gathering. So you have a biological question. So we usually do transcriptome analysis studies with starting with a question. So you have a biological question that you want to know, and then from that question you will craft an experimental design, and then you isolate your RNA. So depending on what method you use, you have either RNAseq or microarray. So these are the general methodologies for them. For microarray, you have labeling, hybridizations, then image analysis because you are looking at the fluorescent tags on the chips, and then normalize it because again this is microarray is heavily statistically reliant because of course it's a comparison between the two signals that you have. And then you have the RNAseq data. So again, like microarray, you create libraries, but you do sequencing here, and the analysis pertains to the reading or mapping that sequence that you have to the genome, and then you quantitate the expression levels. And then from this data that you have, you can actually do, going back to your research question, you do hypothesis testing, which basically over under express is significantly different or not, something like that. And then you can also do exploratory statistics, you do PCA and clustering data. You can also do classifications, so perform supervised analysis like linear discriminants, supervector machines, etc. Actually, recently I have seen papers that have been using machine learning strategies to actually look at gene expression data analysis. And then from these research methods, you do because these are combination of informatics and wet lab applications, but you also do biological confirmation. So you culture cells, basically in vitro studies, so that you can actually get biological insight, so that you will know the pathway. So perhaps these genes are telling us something, giving us insight on the pathway to this condition, or you can use them as a diagnostic tool for an early disease detection, or this information from them can be used as therapeutics. In therapeutics, so these are the target. We can design drugs that can actually access or can that targets these genes that we have identified, or even the protein encoding the genes that we have identified. So basically, this is the general workflow of what you will do for gene expression analysis. Now, some pitfalls. This is from the a really rather old study, but still applicable today, and it's better to actually learn what we should watch out for in doing these gene expression analysis studies. So controls, first of all, controls for multiple testing were not properly described or performed. So here you have just like any other experimental designs and experimental methods, you must have controls for each testing. So the problem here with the controls in gene expression analysis is what we would define as a normal cell. So that's one of the main problems with the control. So what is the normal cell? And in the microarray, what is the baseline of the normal controls where this is neither overexpressed nor underexpressed? So the tendency for microarray studies is to identify the housekeeping genes. These genes are the ones that are usually expressed in the same amount regardless of the cell type and condition. But how sure are we with those housekeeping genes? So anyway, these are one of the things that are basically being debated. So in class discovery, a correlation was claimed between clusters and clinical outcomes. However, such correlation is spurious because differentially expressed genes were identified and then used to define the clusters. So when you do cluster correlations, so is the correlation erroneous or not? So is a correlation really basically logically sound? So you have this correlation and clustering studies, and that is why sometimes network analysis, the use of the hub genes and the network analysis comes into play here. And then supervised predictions include the estimates of accuracy that were biased because of incorrect cross-validation procedures. So sometimes the procedural issues come into play as well. So that's it for our lectures on gene expression analysis. So thank you for listening until this end.